people in the placebo group. Both the doctors and the patients and the subjects were blinded. Nobody knew which person got placebo and which person got vaccine, but when they unblinded the study, they found no differences in terms of risk behavior. So it wasn't a behavioral issue. They also looked at some of their intermediate study goals. One was to either prevent infection, clearly that didn't happen, and they also looked to see if there was a lower viral set point. The amount of virus in the blood after infection was decreased. And I'll show you a little note about that in a minute. But in general, there was no difference. The people who got HIV infection in the placebo group got just as much virus as the people who got the vaccine, so there was no difference in terms of the initial control of HIV infection. There was clearly more infections in the vaccinees than placebos. This is a dramatic problem for HIV vaccine trials. Imagine you're the next HIV vaccine volunteer, you're signing up for the vaccine trial, and the researcher is trying to convince you to participate when in fact you know, it's been in the paper, that the last vaccine trial caused people to get infected. Are you going to sign up for that? I think people are going to be a little bit hesitant. So there's a huge amount of work to do, in fact, around vaccine trials, and a lot of basic research that actually needs to be done. The lack of efficacy did not appear to be explained by suboptimal immune responses to the vaccine. So people did get T-cell responses. I think that's in this slide. Now, I have to note here, as an immunologist, these are not whopping T-cell responses. You can see here that they did pools. They looked at pools of peptides from the HIV virus, and you can see to the different proteins, the average response was really one pool. Most people only responded to one pool of peptides. Overall, the average was 3. This is probably not going to be effective. If you think about how variable the HIV virus is and how many different pieces of information it can show to the immune system, what we're saying here is that this vaccine caused 3 pieces of HIV to be recognized by the immune system in a virus that starts mutating the minute it hits your body. Recognizing 3 pieces of HIV is not going to be enough. So I think that that's probably one of the main problems here, and what we actually think is that there's going to be a difference between the vaccine epitope. Remember, they picked vanilla as the flavor that they wanted to vaccinate against, and if you're coming in with a different strain of HIV, the epitope, the piece of information presented to the immune system, is going to be different. So you might as well not get vaccinated, because what the immune system is going to see is a different kind of HIV than the one it was trained to recognize. And that's actually what they're thinking in terms of this particular vaccine, possibly that the challenge virus was different enough from the vaccine virus that we will not see infection, and we will not see protection. And that's what I'm trying to point out here, is that when the challenge virus gets into those cells that start presenting the information to the immune system, the T cell's looking around for the vaccine that it was trained to recognize, and it doesn't recognize the HIV infection. So it's not able to protect. How will we know this? We'll know this by studying the people who were somewhat protected. And this actually shows that in the vaccine, in the vaccine group, there was actually a number of people who had very low viral loads after infection. So they were somehow able to contain the infection. That tells us that perhaps their T cells actually recognize the challenge virus that they were challenged with. So they're going to look at their T cell responses, they're going to sequence the virus that they were infected with, and they're going to compare that to the vaccine strain. I will bet anyone in this room lunch that the epitopes in the vaccine are the same as the HIV that they were infected with, and therefore they were able to control the infection. But we know this. We already know that HIV mutates, that 3 T cell responses is not sufficient. Why do we go forward with this kind of study? It's a good question, and I'm going to have Pat Thomas get up here and answer the questions with me when we go to the question section. So what's going to happen now? Basically, most of the vaccine trials have been put on hold. We're going to try and figure out, this is what they're doing with these subjects who are exposed to HIV. They're going to look at their type of T-cell responses. They're going to look at how the T-cells responded to the HIV infection. They're going to look at the different routes of exposure. Was a certain sexual practice more likely to cause exposure? There is some discussion, actually, that circumcision might have played a role here. They think that the people who were more likely to get infected were uncircumcised men, and that certainly played out in Africa. We know that circumcision can protect against HIV infection, so they're going to start looking at that, and they're going to stratify future vaccine studies by circumcision status, which makes it very complicated for HIV vaccine researchers. They're going to sequence the virus, and they're going to follow the HIV-positive patients to see if their vaccine had any impact on the course of their disease. If you remember what I said, that is one possible endpoint for a vaccine program. There's lots more to do. They need to look at the set points. They need to look at the immune correlates of protection. They need to figure out if the breadth of T-cell response, which is what we would argue is critical, was important in terms of the patients who were able to actually control their initial infection. Meanwhile, what is the vaccine research world going to do? So basically, if you were working on an adenovirus vaccine for HIV, you're dead in the water. You might as well forget that program, put your R01 grant on the shelf, and start working on something else, because that's not going to get funded, nor is it going to get into trials now. People are looking at alternative ways of delivering the HIV information, either by a DNA vaccine, which we can talk about, or the smallpox vaccine. The pox vaccinia virus could be a potential means of delivering this information. So people are now looking at those potential alternatives. The 3 vaccine trials here, if you go on the website, which is what I did a couple days ago to get this information, the 3 adenovirus vaccine trials that were in process, including one funded by a consortium of donors, including the Europe and the CDC, called the PAVE trial. All adenovirus-directed vaccines are dead in the water. The vaccine trials have been canceled. The people who were recruited to participate basically being told that right now we cannot go forward. Now, the list of potential candidates is pretty huge. What I want to point out about this list, in this huge list of vaccines that are actually in the pipeline, is that they are all vanilla vaccines. They are all basically based on whole protein. They're all basically based on delivering a whole protein to the immune system and never really focused on the variability of the HIV virus and some way of stimulating a broad immune response against all the different flavors of HIV. There are 3 vaccines that are pointed to with arrows on this slide where they're actually considering that, but again, they're using 1 or 2 or 3 epitopes, which really would not be sufficient to protect. One is the Wyeth vaccine. They're using 4 CTL epitopes, and they seem to think that that might be effective, and I really doubt that. So here's the issue that I have and a lot of scientific researchers have with the vaccine development field so far, and that is HIV is a global problem. HIV doesn't just exist in the backyard of the Merck vaccine company where only the clade B flavor is circulating. HIV is incredibly variable, and we really need to think outside the box and how best to address this problem. So that's actually something that we've been working on since about 1996 at Brown, and that's what I'm calling a new vision. I'm not the only researcher working in this area, and this is kind of what we're hoping to develop. We want to develop an HIV vaccine that's effective everywhere in the world, where all the different flavors of HIV occur. We want to make sure that we induce a broad T-cell response, not something that just